Well, then I will start with the introduction of my PhD that is entitled Ecology of the Mountain Treeshrews, the Tupaia montana mainly, along an altitudinal gradient. Why altitudinal gradients? This is because altitudinal gradients represent a shift in environmental variables as temperature and moisture, and because of that there's a shift in the ecology of the communities, and also, well, it represents a very good scenario to study evolutionary and ecological studies. This is our study area. It's located in Borneo. In this map can be seen the transects of the two tallest mountains of Borneo. The lowland locality is Poring, and is about 300 meters, and the highest locality is in Kinabalu, and is about 3,000 meters. And here were done two field campaigns, and we have a lot of very good data of those two field campaigns, and also a lot of specimens which we work with. With altitude, there's a change in the trophic ecology of the communities that we study, also with the parasites community, and parasites community and trophic ecology were correlate, and parasite communities correlate with the immune system, and the immune system at the same time in turn is correlate with the parasite community. We will study Tupaia montana mainly. I will show you a video of these animals. It's worth just to show how were these animals, because we're not very well known. They were an order of Scadentia and were related to primates, and it's supposed to appear soon, I hope. Well, meanwhile, the video has to show how Tupaia are feeding on nepenthes or pitcher plants that were carnivorous plants, that they were feeding on that nectar, and at the same time use those plants as toilet, because it has a big recipient where the Tupaias defecate inside and then give to the plant the nitrogen that they need. This is the pitcher plant. It's a commensal of the Tupaia, different species of Tupaia, and also Ratus valuensis. We will study other species of Tupaia, also mainly Tupaia montana, but also some of their relatives, which were Tupaia minor, Tana, and Longipes. And my PhD will be structured with at least 4 chapters, and I will start with the first one, that is about the trophic ecology of Tupaia montana through an altitudinal gradient. Through the altitudinal gradient, we expect that will be changes in the trophic ecology with the trophic niche and trophic width of the Tupaias. Mainly, we expect that with altitude, at high altitudes, we will have wider trophic niche and maybe a higher trophic position, because there's less competition. And respect sex, we know that females, well, between two sexes there's different nutritional requirements, because females with lactating and pregnant process, they need usually more nutritional input, and maybe there's a difference among the two sexes. And we want to test also if through time there's difference, because even on tropics there's no very well marked seasons. But anyway, we study a very complex ecosystem, and maybe because there are some plants that fructify at some determinate periods, we want to know if through time, there's changes in the trophic ecology of those animals. And we will study this trophic ecology through stable isotopes analysis. We will study the carbon isotopic signature, because among plants, they have different photosynthetic pathways, and then the carbon signature is maintained through the trophic chain. Then we will study the feeding sources by mean carbons. And by mean nitrogen, we will study the trophic position of the Tupaia, because with nitrogen, at each step of the trophic chain, the nitrogen increases. Then, for example, a Tupaia that is feeding on a carnivorous coleoptera will have a higher trophic position than other species of Tupaia that were feeding on herbivorous coleoptera, or other Tupaia that were feeding on plants or fruits. And we also study different tissues, because it represents a temporal window. Each tissue represents the time when the tissue has been synthesized. Then liver has a very fast turnover and represents a week of the diet of the animals. Meanwhile, hair and muscle represents some month of the diet of those animals. And here, there's the first analysis of the carbon isotopic signature through altitude. There's a significant relation, and it increases linearly with altitude. Respect nitrogen, we have a U-shaped pattern, and the model in that case is a quadratic model, and well, it's also significant. Respect sex, we have a almost complete overlap respect carbon isotopes, and with nitrogen, males have a bit higher position than females, but the overlap is also complete and there's no significant difference. With respect sex, the models were done by means linear mixed model, and the random variable is altitude and location, because we want to extract all the variance due to that. Respect time with carbon, we find difference among the different tissues, especially hair is much different of liver and muscle, and respect nitrogen, liver has a greater values of nitrogen, and with those results we can conclude that there's a trophic shift in diet with time. I will go to the second chapter of my PhD that will be about the Major Histocompatibility Complex in all Kinabalu Tupaia for all the species that we have samples. And well, first of all, I will introduce to all of you the MHC molecule that is a set of cell surface proteins that mediate for the immune system, and with the leukocyte cells, which are immune cells. And we have two classes, class 1 that mediates for the cellular immune response, and the class 2 that mediates for the acquired immunity or adaptive immunity. And well, the question if all different species of Tupaia have similar histories in the selective pressures of the MHC. And for that reason, we will sequence all the Tupaias that we have, and we will take also data available from GenBank that there's data from other species, which is a model species that is Tupaia belangeri, and we will compare selection across the phylogeny. And well, these were the first results that we have at lab. That, well, here the first qPCR shows that through the different Tupaias, the first primer work differently. That means that we have to do different PCRs per each species, and with the second pair of primers, at least this one with only one PCR will be enough. But now we are optimizing the protocol in order to know which is the best, well, the best amount of DNA and the profile that we have to take for each PCR and avoid the chimeric sequences. And the third chapter of my PhD is about the Major Histocompatibility Complex. And we will relate along the elevational gradient with treeshrews, and we will, well, see as with altitude, the mammal community, especially the treeshrews, changes as also vectors and parasites. All those vectors and mammals were intercorrelate with parasites, and parasites in turn were correlate to immune system, and this immune system affects also the parasite that the animals has. Then we want to test if at different altitudes the allele frequencies of the MHC were correlate to some kind of immunity or maybe susceptibility to some kind of parasites. And also we want to know if the allelic diversity is correlated to the parasite diversity, the allelic diversity of the MHC. And well, in order to do that, we have the parasites data of some of the animals, and this is a collaboration with people who are parasitologists from the Smithsonian, and they will do the identification by means morphology, and well, we will genotype by NGS all those MHC loci, and also we will do the lab work with the parasites. We will do the identification by means of barcoding of the cytochrome oxidase 1. And well, for that moment, we find different species of parasites, some different species of nematoda, where all those were really different because it's a very diverse group. We have also trematodas and cestodas and also some acanthocephala. And well, trematodas have snails as intermediate host. They were transferred by food. Nematoda were everywhere and can be found at soil, plants, fruits, and the most part of animals has nematodas inside. And well, we will pass to the last chapter of my PhD that will be about the interaction with the MHC and the parasite diversity respect the summit rat, which is the Rattus valuensis. As you can see here, the rat is feeding on the nectar of this pitcher plant. Then it behaves as the Tupaia, but Rattus feeds on those plants during night, while Tupaias feeds on the same plants during day. Then those animals share the same habitat and the same, well, present the same behavior, but were not correlated with taxonomically, were very different groups. And also because Miguel Camacho, Irene Quintanilla, and Jen study the population genetic of those rats, we know that between the two mountains, there's two different populations completely isolated, and within those two very separate populations with a genetic flow, there's a strong drift, and we want to know if this variation also affects the functional MHC genes. And we also at the same time want to know if the MHC alleles, the diversity, is related to the parasite load and abundance of all of the two different populations. And well, to do that, we did the same, the dissections of those rats with the identification of the intestinal parasites, and we will do NIG also with the next generation sequencing with the MHC genes. And well, I'm especially in depth with Jen Leonard, who helped me a lot with everything, and also with Miguel Camacho, who goes to field and take all the data. Also to Arlo, because he goes this year also to field, and we look for salamanders altogether. To Giovanni, because he makes my weekends at EBD much better. And especially to the Irene Quintanilla and Ana Cornellas, who helped me a lot with the lab work. And well, they teach me also, and were both very good teachers. To Gregorio, who stands for a lot of trials of that presentation, also my family. And well, I'm also grateful to Inés and to Carlas that also helped me with his comments, with their comments. And to all of you for your attention. And that's it.